We're going to get started. My name is Holly Lee. I'm the Director of Teaching and Learning. Welcome to our Spring 2019 Undergrad Research Symposium presentations. Before our student presentation, I would like to invite Dr. Chris Gray, Vice President of Academic Affairs and Workforce Development.
Thank you, Dr. Lee. So I'm a storyteller. This is my area. So I want you to think about the baseball as everything in the world that we would ever know, right? So when you went to grade school, or even before grade school, your parents started building on that. They put a little band and you wrapped that really tight with rubber. You wrapped it with things like learn your ABCs, learn manners, learn when there's someone comes threatening after you, hide behind your mom, right? We learn those things. And then you go into formal education, we start to add on to that, right? And so when you get to high school, right, you think, I've got it, I've learned the world. The reality is you've got about half of the baseball core, right? And then you go to college and you start to realize, holy cow, there's so much more in that everything we know about knowledge that you never even knew existed, right? One of the things I've loved about college is my worldview went from here to here overnight. I had no idea that those things in the world even existed. Well, the tools you're learning in this undergraduate research symposium are actually, you're getting outside of that core, that baseball. These are the tools that very, very slowly expand that baseball every year, right? So when Dr. Lee finished her formal education, what it means to be as a doctor means you took the edge of that baseball and you pushed it just a little bit with some new knowledge, right? You're changing that. What you're learning right now are the tools. What you're going to demonstrate to us is you're starting to understand that world and those tools. So I want you all to come back in 10 or 15 years when we can introduce you as Dr. Whoever and show us how you've changed the world. Guys, this is one of my favorite events here at the college. I love nerdiness, and this is nerdiness personified. So congratulations. Unfortunately, there's another meeting where most of us are. Some administrators are going to leave. That's why we're recording, because we want to see the work you're doing and celebrate it, and I can't wait so we can display the work you're doing. So congratulations on the good research you've done, even if your findings did not show any significance or show that we've added to any knowledge base, even if they didn't correlate with all the other body of evidence that we know, you still have learned the tools that will help to expand everything we know in the world. Good luck and thanks.
Thank you, Dr. Gray. Our first presenter is Marissa Blakely and advisors Marla Garrison. Marla, would you like to introduce?
I would love to introduce Marissa. She doesn't get a chance yet until I get to tell you about Marissa. So Marissa came to me at the end of, fall semester, and she said, I've got an idea, and I would love to do some undergraduate research with you, and I want to figure this problem out. And up to this point, I'd worked with 19 other undergraduate students, and normally they come to me and they say, I'd like to do undergraduate students, what do you think, or I want to do undergraduate research, what do you think I should do? Marissa came and said, I want to do this, I need you to give me what the funding and support to do it. So she already had made her general observations, had picked up little tidbits of information from her observations at where she worked, and she was applying content knowledge that she had learned fall semester in microbiology to this question, and she just, by golly, wanted it answered. And I will let her introduce the topic. Marissa, first, before we begin though, tell them where you're going and what you're going to do.
So I want to become a nurse, and I want to go into the nursing program here.
So she is going to be applying for a nursing program here, and then she'll do the bridge to finish her BSN. And knowing Marissa, that is not something she's going to stop at. She will go on and get her MSN or DNP, I know, someday, especially the Doctor of Nurse Practitioner, because she loves research and she has the mind for it. I wanted to tell you that Marissa was so enthusiastic about this project. This project did contribute to the body of knowledge, as did Sydney's, as did Luca's. So when we're talking about that baseball that Dr. Gray just mentioned, they have already pushed it out beyond just the tools, and their 3 projects that you're going to hear today absolutely contributed to the body of knowledge. And Marissa's project was complete from start to finish. We are looking to publish that with the Department of Health in McHenry County. We need to contact them and get it at least in the newsletter. And to tell you that Marissa gave such an unbelievable interview with the Argonne National Laboratory judges this past Friday and the Fermilab scientists that were here judging, that she won first place in the Skyway STEM competition out of all the biology posters, and she is the proud new owner of a blue ribbon for first place. And so this project rocked the Argonne scholars and fellows to the core. They were completely impressed. We will be getting back their impressions later in written form, and Holly will pass them on, I'm sure, to me, and you'll be able to read what nanoparticle physicists and neutrino accelerator physicists and biochemists have to say about Marissa. So I just want to let you guys know that what you're about ready to hear from Marissa and Luca and Sydney is true science.
Okay, so when you go bowling, the first thing that you're concerned about is the bowling shoes. Do they smell? Do they have fungus or bacteria in them? And are they disinfected properly? And that's actually my job at a bowling alley, is to disinfect the shoe rentals, among other things. And after taking microbiology, I was more aware of my environment, and I started to notice the bowlers using the public bowling balls. Then they were eating finger food, going to the bathroom, coming back and bowling again. And I thought that they had a potential to be vectors of disease transmission. So I looked into it more, and I couldn't find any products, protocols, or research done on them. And that's when I went to Marla with my idea. So we decided to search for Escherichia coli, which you may recognize as E. coli, and that's an indicator of fecal contamination. And this means that other pathogens being excreted from the body through feces are likely to be on that surface as well. And then we also searched for Staphylococcus aureus, which is a opportunistic pathogen that colonizes about 30% of the adult population on their skin and in their nose. And this is a potential problem because if they're on that surface and they gain entry into the body, they can infect any part of the body. So we went in on a Saturday night, which is the busiest night of the week, and we swabbed 168 bowling balls. And for E. coli, we swabbed each finger hole of each bowling ball with a cotton swab and sterile saline. And for E. coli, we put it into lactose broth, which is a presumptive test. The E. coli ferments the lactose, creating gas, which you can see the bubble in the test tube right there. That's an inverted Durham tube, which is a super small test tube, so that it trapped the gas and we could see the presumptive results. So there were 19 presumptive results, and it is also a enrichment broth, which means that the few cells that I did capture turned into billions. And my next step to confirm this was to transfer it onto eosin methylene blue agar, which is a selective differential agar. This means that it can narrow it down, and then it narrows it down even more. So it's selective because only gram-negative, which is one of two major categories of bacteria, can grow on this agar, and then it's differential because only E. coli shows up as that metallic green that you see on the top and on the bottom. And so I had two positive tests for this. So 2 bowling balls had fecal contamination, which is too many. And then for Staphylococcus aureus, I followed the same protocol that I did for E. coli, except instead of putting it onto lactose broth for the presumptive test, I put it onto mannitol salt agar, which is also a selective differential media. It's selective because of the salt concentration in the agar. It prevents the gram-negative from growing. And it is differential because Staph aureus ferments the mannitol in the agar, creating a acid product which changes the pH indicator yellow. So that's why you see those yellow regions at the top. That's a positive presumptive test, and then the bottom is a negative presumptive test. And the next step was to look for the colonies that match the morphology of Staph aureus, which is golden and perfectly circular, and that's what you see circled on there. So we took those colonies and we put them into rabbit plasma. And the reason I did this was because the Staph aureus produces a protein called coagulase, and coagulase causes fibrin formation, which is a clot. So, and this is one of its virulence factors. It's what makes it dangerous. And I got 57 clots. So about a third of the bowling balls that I tested had Staphylococcus aureus on them. So you can see all the results here. And this means that bowling balls are commonly contaminated with Staph aureus and can be contaminated with fecal contamination, meaning that they are vectors of disease transmission. However, you can still go bowling. There are steps that you can take. So one that I recommend that the bowling alley can do and bowlers can also do is use disinfectant wipes to clean out the finger holes of the bowling balls. And then to increase that, especially if you're eating the finger food, which is what bowling alleys primarily serve, the only thing that we have on our menu that doesn't require your fingers is a salad. So you can use hand sanitizer when you're going from bowling to eating your food, or utensils to further distance yourself and decrease your risk. I did talk to my manager about this, and he is putting out more hand sanitizers, especially for kids' birthday parties. And he's possibly sending my research to corporate. So, have fun bowling.
What great research. Even I'm a non-scientist, I learned something new because I oftentimes take my kids go bowling. So this is so practical how it affects us. Thank you, Marissa, for such a wonderful job.
Thank you.
Please take it. Do you have any questions for Marissa before she sits down? So appreciate.
Oh my goodness. Thank you.
There'll be some time later on, after every presentation, we have the poster, and we may also stay for questions. Our second presenter is Luca Palacios. Thank you. So his advisor, Dr. Cheryl Cordick, is not here today.
Couldn't make it today, unfortunately.
So would you please elaborate on the research for us, because it's totally off my league. I could
Sure.
not understand, but it
Absolutely.
sounds really amazing.
Thank you.
Oh, and can I just speak up about Luca too very quickly, because although I'm not his
Please,
advisor,
that is
since
here.
his advisor isn't here, Luca had more people at his poster than I've ever seen at any competition. I've done this for a decade. Everybody came by and spoke to Luca, and Luca stood there, and he was the essence of confidence and competence. He answered every question to a depth and detail that you would see in a graduate program at any university, and impressed all. He received a second place medal in chemistry, and he has done this chemistry department at this college very proud.
Thank you. So the research that I have done with Cheryl and my partner Grace Jiu Wang is primarily about drug research. So our project is a demonstration of how computational chemistry can be applied in the drug discovery phase. So drug discovery typically takes 10 to 15 years from start to finish, and we're faced with a problem, and that is with cardiovascular disease. You typically can't screen for those side effects early on. And so down the road, you have to go back and take a few steps back and kind of restart the process. So we're proposing that computational chemistry can be used as a preliminary method to detect possible cardiovascular side effects and save time and money down the road. Do you mind if I?
Yes, please.
So like I said, in 2016, my research partner and our project supervisor discovered a preliminary trend that correlated log p values to an increased probability of CV side effects. Now log p stands for the logarithm of the partition coefficient, and that describes how a drug partitions between water and octanol, and this simulates physiological conditions. So with log p, we're able to get a basic understanding of how lipophilic a drug is and how the drug might move throughout the body. And so my project is an expansion of the original data set, as well as a further analysis of all 61 drugs included in the study. And so the drugs in the study are broken up into 4 groups. In our experimental group, we have drugs that are currently on the market with cardiovascular side effects. We have non-steroidal anti-inflammatory drugs, also known as NSAIDs. We have drugs that have been withdrawn from the market due to cardiovascular side effects. And lastly, in our control group, we have drugs that exhibit cardiovascular side effects in less than or equal to 1% of their clinical trial patients. And so in expanding the data set, I was able to confirm the original log-P trend, and in addition, I did a further analysis and found out why these drugs might be exhibiting these side effects. And to talk a little bit about the process. So I used a program called Spartan 14, which is a commercial computational chemistry program, and I created 3D models of all 61 used in the studies. And I used 2D models as reference to create the 3D models. And after creating models of those 61 drugs, I ran calculations using the Hartree-Fock 321G method, which would give me a series of parameters that relate to biological activity. And so in doing so, I confirmed the original log-P trend, and I also took a step back and I looked at the ADME reports, I looked at toxicology reports, and I found that QT prolongation, also known as delayed ventricular repolarization, was a common side effect in our experimental group, especially the drugs that have been withdrawn from the market. And so I hit sort of a dead end there. So I took a step back and I looked at just the structures themselves, and I found that in our experimental group, there was an overwhelming presence of tertiary amines, which is a nitrogen atom with 3 bonds and a lone pair. And what was significant is that these molecules had long carbon arms and a tertiary amine in the middle, which is consistent with our finding in log-P because they're very lipophilic. And so I was able to cross-reference literature, and I found out that the tertiary amines found in these drugs could actually cause blockages in the potassium ion channel, which would result in side effects like QT prolongation and arrhythmia. So in using computational chemistry, we were able to find a red flag that would constitute further screening in these drugs. And with further research, we were able to cross-reference literature to find out why these drugs exhibit these side effects. So we propose that computational chemistry can be used as a preliminary screening method in early drug discovery to save time and money down the road. Thank you.
Luke, are you going to publish that?
I'm sorry?
Are you going to publish that?
Yes, we submitted for publication in the Journal of Computational Chemistry.
And where are you headed next year?
I'm headed to DePaul to study computer science with a minor in chemistry.
Great. Our next one, presentation to Sydney Rowe, and also the advisors of Marla Garrison. Marla.
So I have to hop up again.
Please.
And tell you about Sydney. So first of all, she also took microbiology in the fall semester, and she also approached me at the end of the semester and said, I want to do some microbiology research because I want to go into genetics and microbiology. And I'm a molecular microbiologist, and so it was like finding clay that I could mold on a potter's wheel. And it was just so lovely to hear a young woman say to me in 2018 in the fall, I want to go into genetics and microbiology. And that is not something that we very often hear at this school. And Sydney has the mind, she has the skill, and she has the observational abilities to do just that. And so I assume, in fact, I demand that in 6 years she have her PhD from now. And if she thinks I'm not going to track her, she's wrong. She's going on to the University of Iowa. Sydney's project is true science. And you can tell by Lucas, you can tell by Marissa's, and you will tell by Sydney's. We have 3 first class winners here. They are the most articulate, which apparently I'm not, articulate students that we have. And they articulate in an explanatory way. They speak so well that you know they understand their material. And Sydney can tell you down to the gene operon transcription factor quorum sensing level, the signaling pathway for her research. And she had read an article that came out in Nature in November last year. It was a premiere article that she's going to tell you about, so I won't steal the show here or give a spoiler review. But she read that article and she was extremely intrigued by it. And she had been working in microbiology with both of the organisms that were in this article. And she wanted to follow through on something the article claimed would be a potential possibility in pharmaceuticals. They were really stretching their results at the end of this Nature article. And she wanted to see whether or not what their claim was would actually be possible. And so she did a research based on that. I want to say that a hero of mine is Louis Pasteur. And he was a French chemist and the first real true microbiologist. And a quote that I always teach my students that Louis Pasteur made was that in the field of observation, chance favors only the prepared mind. And the 3 young people you are listening to today have prepared their minds so well that observation and their education and their knowledge base has allowed them to complete these projects and complete them with incredible panache. And Sydney did not have the opportunity to extend her research beyond the end of this semester. And she, her particular research took a lot of troubleshooting. And we only had 1 semester to do it. If she was going to be here another 2 years, she would have completed a master's level undergraduate research thesis with what she has begun. Sydney Rowe.
Emailed my PowerPoint to you.
Did you email to Tracy?
It was T Champion.
Yeah, Tracy. She emailed to you. So you don't have it?
I emailed it to you.
You can check your sent.
I'll do that. Thanks, Marissa.
Are you logged in as yourself there?
No, I think it's IT logged in. You can go to.
Can I just log out?
Yes. You can just go to, is in my MCC email or the Gmail?
It's a personal one. I can just, I'll figure it out. Sorry. Okay, so now that that's figured out. My name's Sydney Rowe, and before I start, I'm going to throw out a lot of terms, as Marla mentioned. It is an in-depth research project that I did, but just bear with me, and I promise I'll explain it the best I possibly can. Don't get overwhelmed from the first sentence I say, because that tends to happen, and I can see people's eyes glaze over. So please just bear with me. So my research project, as Marla stated, was based off of a recently published Nature article, which found that administering Bacillus subtilis as a probiotic effectively decolonized Staphylococcus aureus in carriers. Now, before I continue, most people are asking now, why does that interest you? So I'd like to throw kind of an idea into your head. Our society is becoming more of a concrete jungle, as we hear. Our kids play on concrete playgrounds. We constantly clean everything. I know my mom probably has 20 cleaning products sitting on our counter alone. So what this article is kind of suggesting is that might be leading towards something that we're not prepared to handle. Bacillus subtilis, as was mentioned before, is a common bacteria that's found in soil. We ingest it when we eat vegetables and all this other things. So by our society leaning more towards concrete and constant disinfection of everything we touch, we might be affecting our normal flora, which is just bacteria commonly found on our skin. And thus we might be gearing ourselves towards more problems, more intense things that we're not prepared to handle, like I'll get into. So this article suggested that the reason behind this decolonization of Staphylococcus aureus is from a cyclic lipopeptide produced by Bacillus species called fengicins. Fengicins in this article were found to have the ability to block the AGR quorum sensing system in staph. The quorum sensing system in staph is just a long chain of reactions that enable staph to release virulence factors when a quorum has been reached. Virulence factors are just things that make staph pathogenic to us when it's encountered on our skin. So this article suggested that fengicins, as released by Bacillus subtilis, were blocking that, leading to staph not be able to release all these virulence factors. So this article suggested that administering Bacillus subtilis could possibly be applied pharmaceutically and prevent staph from releasing a whole slew of virulence factors, which causes it to be such a huge problem in healthcare. Staph leads to intense staph infections, can lead to septicemia. The most common one that people know of is like styes in your eye. That's caused by Staphylococcus aureus. So my research, I wanted to look to see if the primary virulence factor of staph could be affected by this, which is coagulase production. Coagulase just promotes fibrin formation, which causes our plasma to clot, and thus the bacteria is walled off from our immune system, and our white blood cells aren't capable of reaching it, which causes such a big problem. So I had a couple study questions that I needed to investigate. First, whether or not Bacillus species and staph combined could even grow together. And the second, whether or not these fengicins that this article was suggesting could even prevent coagulase production in staph. So the first step I took was performing a growth curve study in which I found that, I'll get back to that later. So my growth curve study, I took Bacillus subtilis and Bacillus cereus, which were just my stock cultures. Bacillus cereus is something that you wouldn't administer pharmaceutically. It actually causes food poisoning, but it was just used to see if Bacillus species and these fengicins could presumably do anything to staph's growth curve. What I found that when I grew staph up in these filtrates, which presumably contain fengicins, that staph was still able to complete its growth curve. Although it may have been affected in extended lag time or doubling time, as you can see, all three of them kind of make the incline shape, which just means that staph was able to keep growing, which is still a big deal if you think about it, because the article kind of suggested otherwise. So the second part of my research study was looking at coagulase production directly. So what I did was I took over-the-counter probiotics that you and I can find anywhere that contain Bacillus species, made that filtrate again, which presumably contained fungicins, and grew staph up in it. I then inoculated something called rabbit plasma, which is just to mimic our plasma in a different way. I inoculated that and found that every single one had clotting in it, which meant that fungicins were not able to block coagulase production, which is what the article was suggesting. So upon further research, which was not mentioned in the article, very strangely enough, coagulase production is not like other virulence factors. Most virulence factors are released when a quorum is achieved, as I stated earlier, and coagulase production is not. What happens for coagulase production is that in early lag phase, which is just the beginning of when a cell's adjusting to its environment, that's when coagulase starts being produced. And then in late exponential growth phase, when a quorum is reached, coagulase production is actually turned off. And this kind of makes sense because the cell wants to wall itself off from our immune system before it can grow and replicate. So what I found is that quorum sensing and coagulase production are kind of an inverse relationship, and thus we wouldn't be able to block coagulase production in the way that this article is suggesting, which is a big deal because if that is true, then we need to really look into coagulase production since it's so different and it's so hard to block it in that case. For example, in my growth curves, the lag phase was about 15 minutes. So if fungicins were if fungicins were administered to block that, you'd have to inoculate a person with them within the first 15 minutes of infection, which is incredibly impossible at this point. So for future studies, after I found that coagulase production wasn't prevented at all, for future studies, I suggested that I had to combine these two studies and at different intervals remove staph and see if at different intervals coagulase production may be affected, even in the slightest.
Do you want to go back and show the quorum sensing?
Okay, so this is a lot. This is what my study is based off of. This is what was found in the article. Basically what you're looking at is the red on top are fungicins, which is what is released by the Bacillus species, and the blue on top is API or auto-inducing peptide. The reason that fungicins are able to block that AGCR protein, which releases this whole slew of virulence factors, is because fungicins and API are structurally similar. So it's kind of like a lock and key mechanism, and fungicins are able to fit into that protein like API would, but instead of inducing this DNA binding protein to start the P2 promoter pathway and release virulence factors, it prevents that. But coagulase production is different. This piece down here, RNA3 is what's responsible for coagulase production, but instead of having this released when a quorum happens, this is started before, and only when a quorum is achieved and those API come into the AGRC protein, that's when this is stopped. So the same thing that expresses coagulase comes back in and represses it. So that's that. It looks like a lot. It's not that bad.
Do you have questions for Cindy?
Yes.
Is it Enviro Medica that did the article?
It's a Nature journal. That's where the article was released.
Oh, okay.
Yeah. It's a lot of scientific articles are published there, so it was also released in...
Well, she's asking who wrote it.
Oh, who wrote it?
Yeah.
Well, where did the article come from? I mean, what, yeah.
You want to describe the study they did because it was not done in this country?
Yeah. So basically the study that was done overseas. It was part of Asia. I can't remember the exact country. Thailand. Thank you. So it was done in Thailand, and what they were doing was they were just looking for nasal cavity colonization of staph, so just where it would regularly live. And what they found was when they looked at intestinal colonization of people through fecal samples, they found that Bacillus subtilis was present in there and that staph was not present in the intestinal wall and the nasal cavity. And the reason behind that is just because Bacillus was able to block staph's ability to cling on to that lining that's in our stomach and our nose, and thus led to decolonization.
Questions? Okay. Thank you, Cindy.
Yeah, no problem. Thank you.
Thank you. All right, this concluded our today's presentation. Just as Marla noted at the beginning, these are true science. I mean, from their presentations today, we see young researchers and future scientists. Thank you so much, Marissa, Luca, and Sydney, for giving us this opportunity to showcase their achievement, to allow us to share the joy of their accomplishment. Thank you. Thank you again for attending, and I would like to, ask you to give them another round of applause for our three researchers and Marla.